we're having a talk by Anirudh Venkatraman, who was a senior in Homestead High School at the time that he did his science fair project. And so Anirudh, I presume you are looking at college now, is that right?
So I'm currently a senior at Homestead High School, so I'm going to be applying in the fall. Yeah.
I
so
see.
I was
Okay,
a junior when I was completed. Yeah.
I see. Okay, so the SFBA ACM was founded in about 1957. Our aim is to promote knowledge of modern computing and create community and support networking among computing professionals and hiring. Our membership is only $20 a month. Sorry, hey, that would be great. No, $20 a year, $20 a year. And if you look on our meetup page, which the link is shown right there, you will see upcoming talks. And we have over 185 past talks as videos on YouTube. We have a YouTube channel, and you can see there again the link for that. We generally have 2 monthly meetings. One is a general computing meeting, typically on the 3rd Wednesday of the month, and that means anything but data science. And then on the 4th Monday of the month, typically, we have a data science special interest group, you could say, talk. During our meetings, certainly when we had in-person meetings before the pandemic, we had networking. We asked people to announce job hiring announcements, and we sometimes had joint meetings with IEEE and Valley ML and other societies. We actually have a meeting coming up in October 24th. I believe it's a meeting on the Ray ecosystem. Let's see. So please take a look at Meetup. Keep looking at Meetup, because we will make other announcements of other meetings as they are set up.
And if you don't mind going back to the previous slide. So folks, if you're here, this is a really wonderful topic. As I just mentioned, I'm actually at the Ray Summit right now in CISO. And one of the things that Ray is really good at is, if you're in the field of data science, it really helps you, take your models and put them into production. And a lot of people who come to SFBay, whether they're in the field of data science or not, they're always interested in figuring out how to actually make AI work right in practice. And Ray is one of those things that it's been really good partnership, as they have done, this will be their 2nd talk for SFBay ACM. And after these series of talks, we are hoping to bring them in for a full workshop. So these talks will actually give you a really good sense of whether or not you want to attend a full workshop and, benefit your career, job prospects, basically develop, a couple of different projects that allow you to showcase, some application with AI capabilities. So definitely, make a strong recommendation for you to attend this talk, and then reach out to any of us through our website, sfbacm.org, and let us know if you're interested in, either volunteering for subsequent talks like these, or for the PDS. We'll come to that in a moment, but again, this is actually part of a series of talks that leads into a workshop in October.
Okay.
Thanks, Bill.
Okay, so I think you were talking about the previous talk that we had on Ray, but you can find that, people can find that on our YouTube channel to see the first talk that we had on Ray, and then another one coming up on October 24th. And the workshop that Yash was mentioning just now was a hands-on multi-hour workshop that we have on a Saturday or Sunday, sometimes both Saturday and Sunday, depending on the needs of the topic. And we provide those for people to upskill in their profession. And we do charge for them, but a lot less than any of the other types of training that you'll see around the Valley. So keep an eye out for that.
Yeah, and for those of you who are present, it would really be great if you can throw a quick note in chat or even use one of those raise hands things or something else to let us know if you're actually interested in the workshop and what aspects of ML pipeline, machine learning pipeline actually interest you the most, because Ray is an open source software. So it doesn't really cost to use anything. That's why we love bringing them on. But we also want to make sure that what they talk about in the entire pipeline is catered to what you guys want to hear about, because we are sort of the nonprofit organization that's trying to bridge the gap in terms of community building and talent building, right? For these critical skills, for the 21st century, so to speak, right? So please let us know through the Q&A or chat, whatever is available to you in this Zoom call. Number one, interested, yes or no? And number two, what aspect of machine learning, deep learning training is of interest to you? And we'll try our best to accommodate that. But please give us some feedback. Yeah.
Okay, thank you. Okay, if you have job announcements or other type events announcements, just put them in the Q&A, since chat doesn't seem to be working again tonight. And here's an abstract of the talk from 2020 to 2022. I embarked on 2 different drug discovery projects. The first project dealt with small molecule drugs using machine learning to expedite processes within computational chemistry. On the second project, I worked on protein therapeutics, particularly nanobodies. Unlike the small molecule drugs, synthetic nanobodies and antibodies are more easily accepted by the body since they mimic our immune system. Discovering effective antibodies remains a time-consuming process, mostly involving extensive wet lab procedures. I was to develop biocomputational techniques to accelerate the determination of nanobodies with higher specificity and affinity to a pathogenic target, reducing costs and time to market, and thereby saving lives. And Andrew, you probably worked with a mentor, a professor at one of the universities, is that correct?
Yes, so for the first project, I did not work with anyone, but for the second project with nanobodies and antibodies, I actually worked with Professor Robert Demoiseau of the UCLA Pharmacology Department. And he advised me to pivot from the field of antibodies to nanobodies, basically.
Okay, great. All righty, and here's a little bit about Anirudh. He's currently a senior at Homestead High School, enjoys music, science, and cooking. And this past year, he won the 4th place at the International Science and Engineering Fair for his project, De Novo Nanobody Design using Neural Networks, AlphaFold, and docking algorithms, in addition to numerous other awards at the state and local levels. Anirudh has a passion for the field of bioinformatics and in particular drug discovery, which he hopes to pursue in college. Apart from science fair competitions, Anirudh performs cell-based research at the Molecular Screening Shared Resource at UCLA under the guidance of Professor Robert Demoiseau. So in his free time, Anirudh likes to play the drums and cook elaborate dishes with his sister and cousin.
Hello, everyone. My name is Anirudh Venkatraman. I'm a senior at Homestead High School. So basically I'm working on college apps right now this semester, and then next year, 2023 in June, I'll be graduating. I've been doing science fairs since about 1st grade. We our middle school, actually, we had a specified science fair competitions every year of elementary school. And then from 6th through 8th grade, I actually entered the science fair competition circuit at the Silicon Valley synopsis. And this is where I sort of was able to progress my love for science fair competitions. And I was able to learn how to compete at a high level, especially at the state level and at international, which I was able to qualify this past year. I was able to actually get started with machine learning pretty much later after I started working on science fairs, actually around the sophomore year of my high school during distance learning. I was able to start some projects with machine learning, but I didn't really grasp the nuances behind it and the linear algebra that goes into every single process. But the accessibility of the algorithms such as TensorFlow and PyTorch sort of drew me in. I was able to complete a few projects regardless of my naivete with regard to the machine learning algorithms. And over the past few years, I've really my love for machine learning has grown a lot. I've been able to perform research with the UCLA laboratory at the California Nanosystems Institute, where I photograph cells, where I'm building a microscope which photographs images of cells. And my algorithm that I developed is able to recognize the cells within those images and segment them to determine what percentage of a petri dish is covered by cells. So, as an introduction to drug discovery is probably one of the biggest issues in the field of bioinformatics. It's 90% of the entire drug development pipeline. There isn't really a whole pharmaceutical industry if there isn't a sustainable way to generate new drugs. And the normal way which most scientists and lab engineers work to generate drugs is through in vitro techniques, which require a significant amount of lab work and it is culpable to human error. And therefore, a lot of the drug discovery techniques are currently shifting to computer-aided drug design. And I will discuss more about that when I get started discussing my first project and then my second project. And I got really interested in the whole field of drug discovery because I'm just fascinated by this idea of distilling so many different variables within how a drug interacts with the body into simple zeros and ones and making this into a language that computers are able to understand. And it's sort of the correspondence between biology and artificial intelligence is what really interests me right now. So let me get started with a quick video demo of my first project that I completed in 2020. So this project had to do with creating small molecule drugs using a recurrent neural network against a certain viral protein. And I will explain more with the slides and with this video.
Hi, my name is Anirudh Venkatraman, and my project is using machine learning to optimize drug discovery and counter viral proteins. Quick notational background, all molecules that I'll be talking about will be represented as small sequences as shown in this slide. For phase 1 of my project, I built a recurrent neural network, which trained on 100,000 different valid and drug-like small sequences from the ChemBL dataset, as shown above. The algorithm that I used was a recurrent neural network. An RNN is a class of neural networks to specifically handle sequential data. And after reading in many different sequences from the dataset, the RNN learns how to predict the next best characters of a small sequence. As you can see, here's my model architecture. It uses LSTM layers, which are long short-term memory cells. For my output, out of 3,000 different molecules that I had generated, 1,859 were both valid and drug-like. Here's an example of generating one molecule. As you can see right here, this is the new molecule that we are generating. For phase 2 of my project, I built a convolutional neural network, which takes in a pair of compounds and their protein targets and is able to output their corresponding binding affinity value. I will quickly demonstrate my model predicting the IC50 score of this small sequence and this FASTA sequence. A FASTA sequence is just a protein represented by a list of its amino acids. As you can see, the IC50 score is around 23 nanometers. The IC50 is how much of the original drug is necessary to reduce the function of the protein by 50%. For phase 3, as shown here, I built an algorithm to edit each of the generated molecules to have them converge to a lower IC50 score. For this, I used a genetic algorithm. It defines an initial population of parents, or in my case, small sequences, and crossbreeds them to create new molecules which would have a lower IC50 value. I generated 4 different drugs for 4 different proteins produced by diseases and compared them to their FDA-approved counterparts. Each of the drugs I had generated had an IC50 score between 0 and 23, and according to research by UNC, the lead optimization of a non-algorithmic drug discovery stage would consider 100 nanometers as the right threshold. Therefore, I can empirically verify that my algorithm produces correct results. Here is a drug my model generated for the protein histone deacetylase, which is produced by a fungal infection. Thank you for watching. The GitHub link is in the description below.
Okay, so that was just a video demo of my project in 2020. Okay, so as I was talking about before, traditional drug development is often costly and time-consuming. And when the root cause of the disease is found, which usually manifests as a viral protein, scientists try to develop lab techniques such as using chemistry, different types of chemistry in order to identify a specific drug that is able to inhibit the disease target by binding to it. But only 1 out of every 5,000 drugs tested on human subjects actually make it to clinical trials. And it takes more than $1 billion to bring a drug from clinical trials to market, which is why I decided to embark on this procedure. Computational chemistry and computational drug development has been utilized by various companies for a very long time. And prediction of the protein shape can also help with identifying the epitope from which you can bind the drug onto the protein. And the advent of these machine learning techniques excited me in a way in order to utilize these algorithms to develop an algorithm that can generate novel drug-like molecules and investigate their effects on the existing proteins to potentially find new drugs for new diseases and paving the way for a much shorter cycle of drug discovery. It will lower costs and accelerate market entry. So this is sort of the flowchart of how my project worked. It consisted of 3 phases: the drug generation phase, the drug validation phase, and then the drug optimization phase. And I tested it against 4 different existing viral proteins, which are produced by a fungal infection, produced by hepatitis C, and then 2 more produced by other 2 infections. And the idea from the diagram here is that the user inputs the protein they want to optimize for. The algorithm generates a significant amount of drug-like molecules at random. It tests all these molecules against the inputted protein to determine how well they bind to it and how well they inhibit it, picks the top 500 with the best score, and then it tweaks each of these top 500 molecules slightly in order to increase the IC50 score or the inhibition score against the protein. And the best molecules are outputted after the score is minimized. And in order to sort of verify if my algorithm worked, I wanted to see if the IC50 value would drop below 50 nanomolars. And that would be my threshold from which I could say my algorithm was successful. So in order to generate drug-like molecules, I used a recurrent neural network with an LSTM cells. And this allowed for the generation of molecules that retain the same semantics as traditional drug-like molecules, because the LSTM RNN was able to learn sequentially how these sequences operate. And in order to encode molecules as sequences, I used the SMILES, or Simplified Molecular Input Line Entry System. What it does is it basically represents each of these elements inside these molecules as letters, and the bonds that form between the elements as special symbols. So you could say a sequence of a carbon chain would be C, 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 C. And if you wanted to do a double bond, you add an equal sign. And this is basically how it represents these pretty complex molecular items as sequences. And let me look at that.
So when you get a second, if you look at the Q&A, you'll see a couple of questions there if you want to take those now.
Sure. Okay. We're able to determine the viability of any of your drug candidates. So I wasn't able to determine the viability through actually synthesizing the drugs as of yet. This project is pretty outdated. It's from 2 years ago. I'm kind of just explaining my original idea and how I, in the next year, sort of focused my project more to make it more nuanced and have it more streamlined. But the overall idea of it makes empirical sense. And it would be a little bit tough to test the viability of my drug because there'll have to be a lot of chemistry performed at very specialized labs, and it gets pretty expensive.
So Anirudh, on a next question, could you click that answer live button, see what that gives us?
Yeah. Yes, so that was my answer to William Paceman's question. Okay. Okay, Joe's question is, so if I get this drug is found. Yes, so the idea is that the paratope and the drug in 3D space, that'll all be latently encoded through all these hidden vectors, because I'm simply taking only sequential data in order to make inferences of binding affinity. So there isn't really much room for paratope variation in other applications as of yet, because it's only dealing with sequential data.
Okay.
And one of the limitations of using SMILES sequences are that it doesn't encode the 3D data, so you don't get some of the dihedral angles that would be sort of useful in order to determine binding affinity. But it was easier to use SMILES sequences because it allowed for a pretty streamlined training process in which I could just use sequential data for both the drug sequences and the protein sequences.
Okay, thanks. I'm
Yeah.
just going to, Joe was saying okay, so I'm just going to dismiss that.
Got it. Okay. Yeah, so the idea is the recurrent neural network is trained, you have a certain amount of molecules generated, and then what happens is that the recurrent neural network is seeded, so it's given a small sample of the training data, and it predicts the next most likely characters until a 137 character string is formed, which constitutes one molecule. And I chose 137 as an arbitrary barrier. It doesn't have to be that. It's just something I chose for this project. Any chance of at least testing your theoretical on simulated array, or is that too far-fetched? So this project, I wasn't really too familiar with all the sort of docking algorithms that were able to be leveraged for this. So I wasn't able to test it on a simulated array, but when I worked on my Nanobodies project, I was able to do a lot more robust testing procedures. So this project mostly deals with the small molecule drugs, and it is sort of my intro to drug discovery as I tried to do it. Yes, I've used the Schrodinger software previously for, I think they have ClusPro for the docking softwares, and I also use PyMol for definitely a lot of data pre-processing. And in order to determine the chemical viability of these molecules, I made sure they followed Lipinski's rule of 5, which is sort of it's a empirical determination of if these drugs are able to be drug-like and valid. And it was implemented through the RDKit Python library. And all of this was able to be trained and run on AWS EC2 instances. So for the second and third phase of the project, I used a convolutional neural network for modeling the protein-ligand interactions because it extracts the low-level features and detects the interactions between the molecule and protein and encodes this in latent space, even though they're both sequential data. And the CNN basically trained on the on a data set of SMILE sequences and a data set of FASTA sequences and the IC50 score between them. So when you have a small molecule drug and it's binding to a protein, the drug is represented as a SMILE sequence and the protein is represented as a FASTA sequence. And a FASTA sequence is just a way of encoding the protein into sequential data. A FASTA sequence just includes all of the protein's amino acids, since that's just what a protein is, a list of amino acids. And you have the initial SMILE sequence, you have the FASTA sequence, and then you have the binding affinity, which is the IC50 score. And what the convolutional neural network does is it's able to extract this data to say, given this drug with this protein, what do I predict the new IC50 score is going to be? So that was how I trained the convolutional neural network for the IC50 score model. And then as for the converging to the best drug for a specific protein, I used a genetic algorithm where I populated an initial set with SMILE sequences that were generated from the RNN, and I wanted to optimize for the lowest IC50 score. So what I did was I took all of these SMILE sequences, I got their molecular fingerprint, and I broke them up according to their bonds and randomly recombined some of these molecules together in order to create sort of hybrid molecules. So you'd have a parent, you'd break the bonds, and then you have another parent break the bonds and combine them to form children, which have equal parts of both parents. And you take the split ups from the parents, which have the highest IC50, and use that to optimize for the highest affinity small molecule drug. So this is basically just how I did the molecule generation. This is what SMILE sequences actually look like, and this is what they look like as an actual compound. And it was trained for over 200 epochs on 8 GPUs. And this just shows a sample of 10 generated compounds. And each of them have been tested for being drug-like and chemically valid. So another thing I'd like to show is the synthetic accessibility of these generated molecules, which just says how hard are these to actually make in a lab. And they mostly range relatively easy due to the fact that the compounds that these molecules were trained on actually were in vitro compounds, and they weren't too hard to generate.
Anirudh?
Because they come from a library. Yes.
You mentioned just now in your previous slide, I think, that you had 8 GPUs and 200, or was it 2,000 epochs, something like that.
200 epochs, yeah.
Excuse me? 200
yes, sorry. Yeah.
And that took around 12 hours.
Mm-hmm.
How big were these epochs? I mean, how much data was processed?
Yes, so it was a pretty robust neural network. The architecture, it was 128 LSTM by 256 by 512. So there were a significant amount of layers, since there is a lot of depth to these, to the training data. There's a lot of features in terms of the bonds, the compounds, the just everything about sort of these SMILE sequences.
Okay, that sounds, yeah. How many data points were in your data set for training data set?
So there was 100,000 different
Okay, all right.
sequences. Yes,
Thanks.
no worries. And as for the IC50 prediction, the convolutional neural network was able to converge to a 0.41 R squared value on the validation samples, and it's a relatively weak. It's an okay correlation, but you can't really converge to anything higher, because there's just so many features that aren't being processed by this network, such as like the 3D structure of the paratope and so on, and sort of like the dipole-dipole interactions that may occur, that just by sequencing, it's a little too naive. So that's why it was relatively low R squared. And this is the final results with the genetic algorithm. Basically, the graph shows the probability distribution between the hybrid molecules versus the value of their parents. And the hybrid molecules are basically the children that were able to that took their recombined parts from the parents. And it was much, much better than the parents' molecules, which means that this sort of optimization actually works, where I'm able to use a genetic algorithm for drug discovery. And this is the comparison between the drugs that my algorithm generated compared to FDA-approved inhibitors of SYK kinase, P14KA, and histone deacetylase, and glutamase kidney isoform. And just for fun, below is a similarity map between one of the compounds and one of the compounds that I generated.
So in the line graph there,
yes,
the bottom axis says percentage better than the average of parent molecules, but those don't look like percentages.
Yes. No, well, these are actually percentages. Like when I'm comparing the IC50 score of the child molecule versus the parent molecule, it was around 2,000% better for a few molecules.
2,000%. Okay.
Yes. So that's basically saying how after many steps of the optimization occurred, it was able to get much, much better over time.
Okay.
Yeah.
Thanks.
No worries. Yeah. And based on my results, I was able to generate molecules that the algorithm considered to counter the 4 existing proteins after 8 generations of mutations. And although my compounds that I generated had a bit higher IC50 score than FDA-approved compounds, I can say that my algorithm architecture works in theory, sort of the idea that the randomly recombining different parts of molecules together in an optimization algorithm is actually working. And I guess some of the problems that I had with this project was, of course, AWS GPU's limitations, and the hybrid molecules may not be chemically valid. So there's a drop off around 2% at each generation of the genetic algorithm for ones that aren't chemically valid. So there is limitations to this algorithm as well. Yeah, and for further research, just sort of gathering more data and running my genetic algorithm longer to converge to lower values, as well as increasing the model complexity to reduce training time with more GPUs. And although it's imperfect, it could play a large role in the field of computational biology. Let's see. I have, for my second project, actually used Google Colab GPUs. I found it much easier, much more simple, with basically an integrated Jupyter notebook with it. So it was a bit more useful. Likely a good model generated. Still ranked as poor. So I actually didn't look at the false positive or false negative scores for this algorithm. I didn't really look into too much of the robust statistics as much as I should have for this project. But the chance is probably pretty high because the algorithm, the IC50 generation algorithm is pretty naive. So it had a relatively low R squared. So that's probably why. Okay. So moving on to my project that I completed in 2021. This is a project on, instead of small molecule drugs, on antibodies and nanobodies. So I started this project because I was talking to my advisor, Professor Robert Demoiseau of UCLA, and he was telling me how companies are not moving in the direction of small molecule drugs anymore due to their imperfect synthetic accessibility, as well as the fact that they have a lot of different off-target binding effects. They're more moving into protein therapeutics, which includes antibody treatments. So stuff like ranibizumab or anything that ends in mab, monoclonal antibody, is a treatment that includes proteins. And I think the main treatment for COVID is actually a monoclonal antibody treatment. Yes, so I'll get to nanobodies in a moment. Let me share the second project that I completed. Okay, so my second project is nanobody design with neural networks, AlphaFold, and docking algorithms. So antibodies are proteins that circulate through the bloodstream, and they tag foreign substances for clearance by the white blood cells. And what the immune system does is they have their own antibody optimization procedure where they create several different of these B lymphocytes, each dedicated to making a new antibody with a different binding region. And this binding region basically tells you how well the antibody will bind to the foreign target. So the antibodies with the highest binding affinities that were generated by these B lymphocytes are kept and allowed to proliferate, while the rest of them are recycled for resources. And the human body serves as a great template for biotechnologists because they actually copy this optimization process when they try to make new antibodies for therapeutic uses. And what they do is they infect bacteria or bacteriophages with unique antibody DNA sequences. And each of these colonies will now act as these factories to produce new antibodies, much like our B lymphocytes. And directed evolution is the process of conducting repeated generations of these factory generation of these antibodies from these colonies of E. coli. And the antibodies with the highest binding affinity are kept, while the rest are discarded, similar to what our body does. So this process has been going on for a long time. It's known as phage display with directed evolution. And I think it was discovered in the 90s. And I think in 2018, the person who discovered it won a Nobel Prize. But it is a little outdated because it does take a lot of money to synthesize these DNA sequences, first of all. And then it takes a lot of time to infect all of these E. coli colonies with these sequences in order to make them generate new proteins. And then you'll also have to take these proteins and determine their binding affinity through an ELISA usually. And just doing this for a bulk amount of sequences takes a long time. Directed evolution. So directed evolution basically is mimicking the process that our immune system goes through in order to find antibodies with high binding affinities against a foreign target that's within our body. And there has been a recent trend in order to shorten this process of using biotechnology to optimize these antibody sequences by using neural networks in order to determine their binding affinity and their sort of variables of influence that affect the binding affinities. And one experiment that did this was Ji Liu of MIT, who used singular neural networks, which treated every variable equally to predict the antibody binding affinity against a foreign target. And I wanted to apply this and make it better. So I decided under advisement from my mentor to work on nanobodies instead. So nanobodies are antibody fragments which don't have light chains or the constant region. So this makes them much smaller. They retain the same binding affinity as antibodies at only 1/10 of the size, which increases their therapeutic capacity and potential utility as a pharmaceutical. And nanobodies are only produced by 2 organisms on Earth, which are sharks and camels. But there are treatments that are emerging, human treatments that are emerging, where they develop these in a lab. And usually what it requires is actually animal sacrifice in order to obtain these nanobodies and sort of edit them through affinity maturation. But if we could generate them in vitro generate them in silico using algorithms, it would eliminate this entire animal sacrifice process as well as shorten the time to market. So the problem was that since March 2020, COVID-19 has taken the lives of 5.6 million, became the biggest pandemic in human history. And as a rise of virus mutations and antibody resistance, nanobodies are actually emerging as an effective counter to these diseases. But the screening and development of nanobodies does take some time and is pretty expensive. So the goal of my project was to accelerate this process using purely machine learning techniques. I would couple ensemble stacking with a gradient ascent optimization process in order to make improvements in the nanobody CDRH3 sequence. And the CDRH3 sequence is the most critical region to binding of antibodies and nanobodies. So editing the CDRH3 sequence can have a very profound effect on the binding affinity of the entire nanobody. So my design criteria was that I wanted to obtain nanobodies with a high binding affinity measured by the change in Gibbs free energy of at least -10 kilocalories per mole. I wanted to make predictions of binding affinity with an R-squared of at least 0.7, a maximum training and prediction time of 96 hours, and cost that shouldn't exceed $1,000. And this all comes back to the project that I completed last year, where I was working with small molecule drugs, the project that I just explained. Basically, nanobodies, unlike these small molecule drugs, they're much more effective in treating diseases because they mimic the immune system response. So when they're added to the body, it doesn't react in the same way as if a small molecule drug was added to the body, where it would have all these emergency reactions going on since there's a foreign substance in the body. And nanobodies are cheaper and can be more efficiently produced in bulk, as well as eliminating animal testing. And a phage display and directed evolution of the primary techniques to perform antibody-nanobody research in vitro. However, these cost millions of dollars. And Ji Liu's paper at MIT of predicting the binding affinity between antibodies and various proteins only yielded an R-square of 0.55 and Pearson correlation of 0.775. And I wanted to see if I could improve upon that. So this is what I did in order to complete this project. Basically, what I had was CDRH3 and enrichment data. So what this data included was a previous phage display experiment. And this experiment had 60,000 different varied CDRH3 sequences. And these sequences all encode the binding region of these nanobodies. And the enrichment is it's a teller of binding affinity. It's another teller of binding affinity that's obtained from these phage display experiments. So that's a significant amount of data to perform machine learning algorithms on. What I did was I split the data into training and testing. I trained a regression ensemble and a classification ensemble. So this ensemble included 6 different neural networks with varying architectures. It included a regular artificial neural network with just 2 dense layers. It included a bunch of convolutional neural networks with various different filters and strides in order to make more nuanced predictions on the data. And once I was able to gather all these enrichment predictions on the testing data, I trained an interpreter network in order to weigh these ensemble predictions such that it's able to take the weighted average of all of these affinity predictions into a much more accurate prediction. And for the second phase of generating optimal nanobodies, what I did was I took the seed data, which included the best nanobody sequences, best CDRH3 sequences with the highest enrichment from the original training data. I performed gradient ascent, so I made edits to these seeds such that it optimizes the binding affinity. And then I ran the enrichment prediction algorithm from this interpreter network from the first phase. I obtained the top 50 nanobodies, and then I performed a simulation using Haddock, the Haddock docking software, in which I was able to determine how well they interact with the antigen in order to determine a good estimate of its binding affinity to see if my algorithm worked heuristically. People talk to you about nanobodies. So nanobodies are much like antibodies in the fact that they treat diseases. They're able to bind to these foreign substances. I don't know if that applies to organ transplants. Maybe some type of drug could be replaced with a antibody or nanobody, such that it's able to the donor is able to agree with the rest of the body, but I haven't really looked into that. And all of this data of the CDRH3 and enrichments was acquired from the previous experiment performed at MIT, Ji Liu's paper on optimizing against the antigen target ranibizumab. So this is what the simulation pipeline looks like. You have the sequence, the CDR3 sequence that was generated from the original algorithm. You take this generation and you fold it using AlphaFold. So it generates the 3D structure of the sequence. And it's docked, which means it finds the best orientation such that it reduces the, it optimizes the release of energy between the bonds. And you are able, from that docked structure, you're able to get a structural prediction of the binding affinity using the intermolecular forces that are necessary for binding affinity prediction. So the ensemble architectures that were used, it consisted of 6 different neural networks with pretty small amount of parameters, only about around 20,000 at max. And this was because we wanted each of these networks to be relatively naive. And why do you ask that? Why is that important? Because if we make it too many layers and too many parameters, that will overfit the data. And the data is very nuanced since it is sequential. So you have the sequence of CDR3, you have a CDRH3 sequence with just a list of amino acids, and you have the you have a protein, you have just like the enrichment data, which is the binding affinity prediction. So if you're performing regression, it's very easy for a model with a lot of parameters to overfit. And the regression networks were trained using the mean squared error loss function and the classification with the binary cross entropy loss function. And these are the different architectures for each of the 6 networks. You had a regular artificial neural network. You had a convolutional neural network with different amount of convolutional filters, as well as strides. And basically what this allowed is that each of these networks would have some sort of unique interpretation of the data. And that would allow each network to, if it's not overfitting, at least gather some sort of nuanced information such that when I'm able to combine these predictions all together, it comes together in a much more nuanced and efficient way. So in order to do this, I trained these neural networks for only 20 epochs, and I optimized their hyperparameters using the tree structured partisan estimator. And in order to implement the interpreter network, I used the process of stacking, in which a single model is constructed, and it combines the prediction of each of these models within the regression networks. And what it does is it takes the outputs, which is just the binding affinity from each of these networks. It feeds them through its nodes, and it comes out with a weighted prediction of how well it weights each of these outputs basically to come out with a more accurate prediction. And it consists of the 18 regression networks and the 18 classification networks predictions of enrichment on an unseen test data set, in addition to the corresponding ground truth value. So it's able to, using the mean squared error loss function, learn how to weigh each of these networks' predictions such that it more accurately predicts the binding affinity of a CDRH3 sequence. Yes, I can. Yes, I can do that. I'll go an overview. Yes, so the next step was the gradient ascent algorithm. And in order to predict new CDRH3 sequences, so ones that it hasn't seen before, ones that the world hasn't seen before, and that could potentially be used to bind to this protein, what I did was I took the initial seed sequences, which had a high binding affinity to the protein target. And what I did was I iterated upon it to maximize the enrichment. So I one-hot encoded all of these CDRH3 sequences. I just it converts the categorical information of these sequences into a vector. And there are only 20 categories because there's only 20 amino acids. And I performed gradient ascent, which basically means that I computed the gradient of the one-hot encoded sequence. And I wanted to optimize this gradient by basically traversing the topological enrichment score function, looking for the highest average enrichment score. So the model calculated the gradient for each amino acid of the seed sequence, and it was multiplied by a learning rate, and it was continuously added together over over a projection interval. And then this continuous representation was converted back from this, from like right here, where it's continuously added together into the one-hot representation. And this would constitute a brand new sequence because you're making edits to the original sequence based on the new continuous representation. So the projected sequences are evaluated for their enrichment by the end of this optimization process. And if it hasn't improved over 10 iterations, it presumes that it has converged to a local maximum. So moving on. For the simulation and validation, each of these sequences that were produced by gradient descent were checked if they meet the biological requirements for folding a valid protein structure. So they had to end in DV or DY, and they couldn't contain a cysteine or an asparagine because they could become glycosylated. And the sequences produced were evaluated by the interpreter network to determine and rank their enrichment. And the top 50 highest enriched sequences were then evaluated through a simulation process. So this involved taking these 50 very highly enriched or very high binding affinity sequences and folding them into their 3D structure using AlphaFold, which I know many of you have probably heard of. Google developed it, the DeepMind team developed it, where they were able to solve the protein folding problem and determine the structure of a protein just by its sequence. And the folded candidates were then docked against the target, which was ranibizumab, using the high ambiguity driven protein docking or haddock protocol, where you took the protein that was folded by AlphaFold and you take the antigen target and you combine them together in a way that releases the most energy. And this is the docked conformation. And it involved identifying the active residues and the regions of the nanobody, the hypervariable regions of the nanobody, as well as the hypervariable regions of ranibizumab. And after docking each of these nanobodies, the highest quality complexes were evaluated for their binding affinity using the CSM antibody protocol, which was developed by Cambridge, in order to make accurate predictions of the Gibbs-free energy, which is able to tell you how well this nanobody binds to the target. Yes, I will also answer that as well. The highest accuracy structure predictions were compared against the highest enriched nanobodies that were derived from the original phage display experiment. So the ones from my seeded training data. And here is sort of the TSNE plot of the training data. And what this tells you is basically there is a algorithmic defined way to find these high affinity nanobodies. Because they high affinity ones, which have a enrichment score of around 1.5 and above, they all cluster in sort of these little areas that tells you that they form a pattern at a TSNE plot. And that indicates the tractable pattern that the neural networks can identify to optimize these CDRH3 sequences. And as for the classification results, it was able to classify positively enriched versus negatively enriched sequences very, very well, with a 96% accuracy and an area under receiving operating characteristic of 0.965. And what this means is it's able to tell binarily good CDRH3 sequences versus bad binding CDRH3 sequences. As for the regression results, as you can see, this is the mean squared error Pearson correlation in R squared for the single neural networks compared to the ensemble of neural networks. So these single neural networks just include these around 15 to 20,000 parameter networks, which are trained on these 60,000 different CDRH3 sequences. And their mean squared error Pearson correlation in R squared is not that great. It's like they're finding some patterns, but it's very unable to act on these patterns and make good nuanced predictions. And that's why when we combine all of these different networks together with the interpreter or the ensemble, it predicts with a much, much more higher accuracy. It has an R squared of around 0.75, a mean squared error of around 0.11, and a Pearson correlation of about 0.87. And this is a predicted versus actual graph of the predicted enrichment versus the ground truth enrichment. As you can see, it follows a relatively linear curve, and the sequences that are positively enriched, they are much more closely gathered together than the sequences that are negatively enriched. And that is because of this previous TSNE plot, which tells you that there are specific patterns to these positively enriched sequences, and that there are certain amino acids that are contributing to these high affinity sequences, but it doesn't really know which ones contribute to these bad sequences. And that's a good thing, because we want to know how can we optimize these sequences such that we only put these good amino acids so that the affinity always goes up. So this is the gradient ascent and simulation results. So this is the optimized enrichment of the nanobodies that I was able to optimize via gradient ascent. And this is actually the seed enrichment. So these are the originally highest enriched sequences from the training data. And as you can see, the optimized enrichment is much higher than the seed enrichments. And that basically validates my entire network, which is telling me that these optimized sequences are much better and much more able to bind to the target than the seed enrichments from my binding affinity prediction. And when I actually docked them, when I folded them, docked them, and determined their binding affinity using this very robust simulation process, my sequences were actually better than these seed sequences. Mine generated a lower delta G of around negative 10.24 kilocalories per mole compared to negative 9.65 kilocalories per mole, which means I could safely say that my algorithm was able to generate nanobody CDRH3 sequences, which have high binding affinities, all purely computationally. So basically, to sum up, the accuracy of the classification networks is 96%. The area under receiving operating characteristic of the classification is 0.965, which means the model is able to very accurately determine high affinity versus low affinity sequences. The R squared of the ensemble is around 0.75, representing a strong linear correlation between the predicted and ground truth binding affinity values. The Pearson correlation is about 0.87, indicating a strong positive correlation. And ensemble stacking produced computational optimized sequences that have a lower change in Gibbs free energy compared to sequences derived in vitro. And if you want to know the nitty-gritty of how Gibbs free energy corresponds with binding affinity, it represents the amount of energy that is used for a particular reaction or operation, and a more negative value indicates that there's a release of more energy. So the more strong bind that occurs, the more release of energy that would happen. And this is what Gibbs free energy basically tells you. And the sequences that had ensemble stacking had a lower Gibbs free energy, which indicated better binding compared to sequences that had that were developed from directed evolution and in vitro processes. So in conclusion, my results were basically twofold. I was able to create an algorithm that was able to determine the binding affinity of these CDRH3 sequences with a much higher accuracy than previously applied. Because G. Liu's paper in 2019 at MIT, he got around an R squared value of 0.55 and a Pearson correlation of 0.775. My ensemble stacking algorithm got an R squared of 0.75 and a Pearson correlation of 0.87. And because my model produced nanobodies with a lower delta G on average compared to the in vitro derived candidates, I consider my approach with ensemble stacking a success. It met my design criteria because it had a minimum delta G value of negative 10 kilocalories per mole. The algorithm predicted with an R squared of 0.75 and only took 36 hours to fully run. And it cost about $900 for the AWS cloud use for the docking algorithm. And for future work, basically what I'm doing actually right now is testing these nanobodies in vitro by synthesizing them and determining their binding affinity against the antigen that I was testing at the California Nanosystems Institute at UCLA. So I'm actually working in the lab in order to determine this process. Very exciting. Be able to actually determine whether this algorithm works fully, and it would be a great way of corroborating my results. So it's currently underway, and I can't wait to see what happens in the future. And these are some of my references as well. All of these algorithms are available on my GitHub. I can link it in the chat as well. So yeah, that is about it for both my projects. In conclusion, some of the future work that I'm doing right now for the science and technology search which is the level above ISEF, is working to generate nanobody sequences using 3D data and sort of the nuances that go with the dihedral angles and these all of these bonds and distances between alpha carbons that would correspond to nuance with 3D structures. So I'm very excited about that. And I was also super excited about my ISEF results from this year, which was I was very happy to receive after completing this project. So yeah, thank you all for listening. Answer some questions.
Yeah, there's a couple of folks are congratulating you on the quality of your work. And William here has an actual question.
Okay. May not have followed entire, but it looks like you did a lot of work. Yes, so that's the entire gist of the algorithm, is that I tested it for one specific target, but it can work on any target. And that is the whole idea of this project, is that it's a case study on this one specific target, but if I were to change the training data, it would work on anything. And that's basically the idea, that you have this one target, you're able to optimize a nanobody sequence for it, and you can just change the target arbitrarily. Does 3D data include rotation bound? Yeah, so it includes, basically what it does is it encodes these nanobody sequences as a graph neural network. Oh yeah, does the 3D data, Joe is asking, does the 3D data include rotations about bonds? So the 3D data, in order for it to leverage 3D data, I construct a graph neural network from these nanobody sequences, where I encode these nanobodies as a graph where the edges have the distance correlations. So it would be an orientation matrix between the residues, and it would also include the distance between alpha carbons and the dihedral angles of the nodes. So there's a lot of different moving parts to it, but yes, it does include rotation about bonds. And Joe's asking, post the GitHub. I will momentarily.
We can also post that on Meetup in the comments.
Yes.
If you like. Okay.
So this is the one for the nanobody experiment, and this is the one.
Okay, so that's going to the panelists, but that it's not going out to everybody. So again, we can post that on the Meetup comments.
Sure.
section.
Okay. Sure. I'll send both. This is the second. Yeah, so if we could just post both of those would be great.
Actually, it does look like there's a way to post to everyone, but that's okay. We'll post it in the Meetup comments anyway. Yep, and on YouTube. Okay. Posted on YouTube as well. All right.
And let me also, I think V.L had a couple questions.
Yes.
It's still there.
That was about 15 or 6, number 15 or 16
Yeah.
in the answered thing, or just
Yeah.
answered
So
thing.
at the end of your slide, he's asking at the end of your slides, can you please go over an overview of how a pharma organization would validate the results of a successful computational drug discovery bioinformatics effort, one level below, noting the about clinical trials. Yes. So in order to validate the results of these drug discovery efforts, what they would do is perform in vitro experimentation, where they would just sort of perform an ELISA protocol where they take the nanobody sequence, or they take the small molecule drug, and they see how well it binds to the target using a fluorescence indicator. And that will tell you how well it binds to the drug. And if it binds pretty well, they can proceed to test it on rats and then proceed to test it on humans. So that's how pharma organizations would usually do it. And all of the data sets that I acquired, they were all public. They're all from papers, and they're all published on the Sequence Read archive. So this is sort of the NCBI archive where they have public data from experiments that were run in the labs. Is there a database? Carl Anderson is asking, is there a database which might help predict side effects? So there are some algorithms I know of which use neural networks to sort of predict off-target binding. But side effects, there's a lot of different variables. There's like infinitely many more than just one compared to binding from two molecules or two proteins together. So that's why it's a little tough. And that's why the whole clinical trials have to take place. That's basically our only solution to it right now, but it's a good idea to look for in the future. Yes, so the cloud expenses were basically funded by my parents, just basically working on AWS, and I was able to get like sort of a student discount as well. They were able to pay for a significant amount of it, Amazon itself. So yeah, that was how I funded it.
Okay. Anyone else have any questions? While people are thinking about that, let me just congratulate you. This is very good work. It looks like very detailed, done as you have told us, carried out over at least a couple of years. I think that's it's very good. I'm sure that this will help you when it comes time to do. I presume you'll be going for something like a PhD, master's or PhD or something like that.
Hopefully, yeah.
We'll see. Yeah. So Carl's asking, conceiving populations for genetic algorithms.
Yeah, co-evolving. So Carl is asking co-evolving populations for genetic algorithms, virus variants versus treatments. So as for the co-evolving populations, I did not use genetic algorithms for my most recent experiment, so I'm not too familiar on the idea of co-evolving populations. But yeah, and for the virus variants, that is more of like a protein folding / protein genomics problem, because we're taking all this data of previous virus variants and sort of predicting how it will mutate over time. So that's not really in the field that I was looking at for these couple projects. And yes, I can post a link to the public data set.
Okay, and again, we can post that on YouTube and on Meetup. Okay.
Yes.
All right. Are you You don't need to post it right this second.
Oh, no, it's pretty. I think I actually in the GitHub repository. Training data is in a Google Drive folder. It's in the README, so you can look at that as well.
Okay, great.
Great. Thank you, guys.
All right. All right. So thanks. And we'll be in touch to try and pass some information between yourself and Joe, for example, or anyone else that needs it. Okay, I'm going to pretty much end this. Thanks everyone for coming, and we'll see you